Okay, we're back. Thank you very much for your patience. I was not feeling well all of a sudden, and yeah, so feeling slightly better. What we're gonna do, we're gonna finish now going through lecture 5. So let me share my screen, which is the introduction to the vascular plants lecture. And we're going to finish it now. We're going to finish it. So we, just to back up a little bit about where we left off before I started to not feel so hot. Yeah, so there's advantages and disadvantages of being vascular. There's advantages and disadvantages of being non-vascular. But in many ways, the advantages of being vascular very much outweigh the disadvantages, right? One big change is that sporophyte generation is now dominant, but as the plants, first start to get vascular, you're not going to have fully developed organs right away, right? You're going to have more primitive roots, kind of primitive stems and leaves, and a more primitive vascular system, right? And no seeds yet, right? We're still working just with the spores, spore-bearing plants. We went through this briefly before, right? This is your example of an intermediate plant from the fossil record. Characteristics are that it doesn't have any leaves or roots, doesn't have, it has stems that are photosynthetic, has rhizoids, and uses mostly a horizontal stem, and the sporangia are located on the stem axes, similar to in the non-vascular plants. But this is an intermediate plant, right? So we have like kind of in between the vascular and the non-vascular. And importantly, we don't have vascular tissue yet, right? We're still working with these leptoids and hydroids. I said earlier, right? Then you start to see the very first vascular plants show up in the fossil record. Same characteristics of that intermediate plant we just looked at, except that now we have a real vascular system developing, and if you did a cross section through the stem, you see primitive xylem and phloem. And we looked at this, showing fossil specimens of sporangia and of a sporangium and spores, right? And then we talked about the slightly later vascular plants, Saudonia here, and they have the same characteristics as the intermediate and the early vascular plant. However, now there's lateral sporangia, which is different. And there's also, so lateral sporangia, which is different. The xylem and phloem tissue is looking a little bit more like what we see in modern plants. And this new term, Enation, which are these spike-like things that come off the stem, which we believe are the precursor to the first leaves, or what eventually turned into the first leaves. All right, so there's a bunch of early vascular plants, meaning plants that evolved around 400 million, or showed up around 400 million years ago, that are still alive today. And that's what that term extant there on the top means. It means that it's something that still exists on Earth, right? And I briefly mentioned these, right? We have the lycopods, which is your Lycopodium and Selaginella, which is shown on the top left and bottom left, and then the horsetails, which we will learn about the lycopods next lecture, and then the horsetails the following lecture. All right, and then this is kind of where we left off, right? This is where we kind of ended up. So vascular plants have tissue systems, and those tissue systems are composed of cells. And those tissue systems are organized, the plant has cells in it. The cells are organized into tissues, and the tissues are organized into tissue systems. This slide should not be this hard for me to get through. And importantly, there are 3 tissue systems that occur in all sporophyte plant organs of the vascular plants, right? Of the vascular plants. Dermal tissue system, ground tissue system, and vascular tissue system. We're not going to spend a ton of time on this today because we will see it again in subsequent lectures, but just so you know, right? We have talked about the process of mitosis before, right? Which is usually occurring to make the plant body get bigger, right? Not for repair, but when you're talking about mitosis being used to make the plant get bigger, this is usually happening at those apical meristems, right? Which is a type of cell that can divide and create new types of cells. So the apical meristem can divide into different cells that then get organized into tissues. Those may, and again, we'll go through this a bunch of times. I don't want you to get too hung up on this today, but basically, apical meristem gives rise to primary meristems, and then those primary meristems give rise to primary tissues. That's what I want you to see here. So 3 types of tissue systems. And those tissue systems are the dermal tissue system, ground tissue system, and vascular tissue system. And the ways that those come about is via this process. Right, okay. Enough about this slide, please. All right, let's talk a little bit about the vascular sporophyte plant. So now we're vascular. So we have real roots, real true roots. They serve to anchor the plant and, as you may know, to absorb water and minerals. Now we have real shoot systems, so real stems, real leaves. The stems are going to basically hold the leaves up so that they can get sunlight and do photosynthesis. And then the vascular system. So this is your xylem and your phloem, right? The water conducts minerals from the roots to the leaves and water. And then the phloem conducts sugar or the products of photosynthesis from the leaves to the rest of the plant. Okay. And then you have sporangia, obviously, because you're talking about a sporophyte plant. So just so you don't get confused, these are tissue systems, right? Dermal tissue system, ground tissue system, vascular tissue system. These are organs. They're still organized into systems, but these are organs: root system, shoot system, vascular system. I know it can be a little confusing because you have vascular tissue system, right? But these are organs here on this slide. And sporangia, of course. Okay. So the tissue systems are going to make up the organs of the plants. That's how they're related. So if you're talking about the dermal tissue system, the dermal tissue system is going to be present on all parts of the plant. So you could think of it as the skin. When you think of the dermal, like we have epidermis, right? Epidermis. So when you think of dermal tissue system, you can think of the skin almost. And on the leaf, so up here, this is showing you a leaf, right? On the stem and in the roots, you will have the dermal tissue system, and that is called the epidermis. So if we were to draw it here on the leaf, so here's the epidermis, dermal tissue system of the leaf. Here's a stem cross section. You have the epidermis around the edges of the stem. And in the root, same thing, right? So that's your dermal tissue system. Then you have the ground tissue system. I'll use a different color. I'll use green. So ground tissue system. In the leaf, the ground tissue system is called mesophyll, right? Mesophyll. And that's basically this whole, the whole inside of the leaf. In the stem and the root, the ground tissue system is called cortex. So that's this whole, and you could draw all the way around, right? And that's in the stem, and here's in the roots. So that's your ground tissue system. And then lastly, your vascular tissue system in the leaf, stem, and roots is xylem and phloem. So I'll use I'll use yellow. So in the leaf, the xylem and phloem is going to be located in what's called veins. Vein. In the stem, the xylem and phloem tissue is going to be in what's called the stele, which is a term that we'll cover in a minute. And in the root, same thing, right? Xylem and phloem tissue located there. Okay. So when you're looking at these early, so notice here it says extant early vascular plants, extant, right? Extant early vascular plants. So this means lycopods and horsetails, right? So Lycopodium, Selaginella, and horsetails. They have more primitive organs compared to plants that evolved later, right? They're a little bit more primitive. Here, and so let's talk about that a little bit. Right, so primitive root system, primitive stems, primitive leaves, primitive vascular system, meaning that the organization of the vascular tissue is kind of basic. So when I say primitive here, I mean basic. I mean like, oops, you're looking at stuff that's like the very beginning of a thing that it then went on for millions of years later and was and evolved and changed. This is kind of like the basic ones, right? More primitive. And if you look at sporangia here, right? Sporangia are located on the leaves of the sporophyte plant. This is a difference than what we saw in the non-vascular plants, right? So you should make a note of this here. Where are the sporangia in these early extant vascular plants, lycopods and horsetails? The sporangia are located on leaves of the sporophyte plant. And some of these plants, out of the lycopods and the horsetails, have one spore type, which we call homosporous. Just to help you understand these terms, homo means same, and then sporous refers to spores. So if you are a homosporous plant, all the spores you make look the same, right? They're all the same type of spore, so there's one spore type. Some of the plants, though, that we're going to talk about in the lycopod lecture are what's called heterosporous, which means there's two spore types. So hetero means different, and then sporous still refers to spores. So if you're a heterosporous plant, you have more than one type of spore, different spores, right? More than one type. Okay. Talk about the root system of these plants. So again, we're talking about the lycopods and the horsetails, right? Lycopods and horsetails in general right now, just like we did for the bryophytes, right? Like first we talked about the bryophytes in general, and then we talked about the true mosses, peat mosses, liverworts and horworts in more detail, right? That's what we're doing here now too. First, lycopods and horsetails in general, and then we'll break it down more. So those root systems, they're very primitive roots, right? Basic. So don't get confused. I'll use green. This thing here is the horizontal stem that we've already encountered. And the roots are actually just these little things. That's it. Not very advanced, right? Very basic, little roots. They're simple. My head is probably covering it, but roots are simple is what that says. And the roots basically will show up along the stem wherever the stem touches the ground. So in these red circles here, you can see like this plant would have been like ripped out of the ground, and these little roots would have been where the plant was touching the surface of the ground. When this is a term we'll encounter in future lectures too, so might as well mention it now. When a root shows up on another part of the plant body that you're not really expecting. Like, so when you think about a tree and you think about where are the roots are like at the bottom of the trunk, right? Like that's where the roots are coming out of. Those we just call roots. But if roots come out of other things, like for example, the stem, we call them adventitious roots, just so you know. You'll probably see that term in the book and also in lab. Roots are very basic, so they follow that same basic growing pattern that we've covered before of that equal dichotomy. That's what this is saying here, dichotomize, equal dichotomy. So as the roots grow, they'll do that grow, split, right? Grow, split, equal dichotomy pattern. These over here haven't split yet, but they will grow and then split, etc. All right, so that's kind of the root system. Oh, and then obviously, right, the root system is discussing the organs, right? And so these organs, the roots, are made up of different tissues that we just covered, the epidermis, and then the cortex, which is that ground tissue system, and then the stele, which is the vascular tissue system. So this word stele, we can't avoid this word stele. It's just going to keep coming up and up over and again. So we got to kind of talk about it briefly, not too much, because we will go into it again in future lectures. So what the heck is this stele? What's the deal with the stele, right? The stele is a term that plant people use to describe how the xylem and phloem are arranged. How the xylem and phloem are arranged inside of the root or arranged inside of the stem. It does not refer to how the xylem and phloem are arranged in a leaf for whatever reason, because leaves, it's like veins. And so we just stop with veins. But in the root system or in the shoot system, when you have the xylem and phloem, how it's arranged in there is what's called the stele. There's 4 types of ways that the xylem and phloem can be arranged or organized. And so they are these. And we'll see these many, many times, so don't be so scared today. There's the protostele, siphonostele with leaf gap, or sorry, with no leaf gaps. So you say siphonostele. Then siphonostele with leaf gaps. And finally, eustele, right? And if you're looking in terms of evolutionary time, protostele is the most basic. Whoops, this is supposed to be an arrow. Protostele is the most basic. So in the Lycopodium and the horsetail, well, not so much horsetail, we'll talk about that, but in Lycopodium, for example, which is a really early plant, really basic plant, it's going to have protostele type of stele arrangement. And notice, look, right, stele, right, so protostele, siphonostele, right, eustele. All right. Yep, this is what I was just saying when you're looking at the slides later, that this is the most basic type of stele organization, and it's what the earliest extant vascular plants have, for example, Lycopodium. When we start to look in the more, I think, a little more complex, Selaginella is going to have this stele organization of siphonostele with no leaf gaps. And then we get a little bit more advanced in the ferns, which we'll cover in a few lectures from now, that have siphonostele with leaf gaps. And then the most recently evolved plants, which are the seed plants, all the seed plants have a eustele. All right. So earliest vascular plants, for example, Lycopodium, have a eustele. And just so you know, right, remember this guy from before, this Zosterophyllophyta, right, the Saudonia, that it has these lateral sporangia and then these little spiky things, the Enations. So just so you know, when we said before that this this fossil plant had more modern xylem and phloem, like xylem and phloem arrangement that looks like it does in modern plants. Well, there you go. If you look at the fossils of Saudonia or Zosterophyllophyta, it also has a protostele. That's what I mean by modern when I said that before. Okay. So that was the root system and a little overview of the stele. Now we talk about the stem system. So stem system is this horizontal stem, which is called a rhizome. Do not confuse rhizome with rhizoid, right? This is something, and I do text box. Horizontal stem is a rhizome. That's a new term for us. Horizontal stem, rhizome. Whereas in the previous lectures, we came across these, I should do it the other way, this other word, rhizoid, right? Which were like hair-like things that anchor the plant to a surface, right? When we were talking about the non-vascular plants, for example. So don't confuse rhizome with rhizoid. There's a lot of words in biology, but particularly in the plant world, that have similar structure, right? Like, for example, bryophytes versus bryophyta, right? Look similar, but they're different words. Likewise, rhizome and rhizoid are two different things. So horizontal stem, it also does that grow, split, equal dichotomy thing that we talked about before, grow, split, right? Oh, I'm moving my thing here. Okay, and because it's an organ, right? The shoot system can consist of organs. They're going to have tissues inside of those organs. So epidermis, cortex, and the stele is going to be in the shoot system. Just to talk a little bit more about what a protostele is, because when you look at that other slide, when you look at this, right, this is not very informative, right? Like this doesn't really give you an idea of what it looks like in a real plant, right? That's because it's a drawing. But if you were to look at a cross-section of a stem under the microscope slide, I'm just trying to see what the difference is between these two. Must be, oh, it's just zoomed in. It's just zoomed in. Okay. Yeah. This is what the stele would look like on a microscope slide. So this whole thing here is the protostele. This whole thing here is the protostele. And inside that protostele, you'll have, here I'll use black for xylem. You have xylem tissue, which is the larger celled tissue, kind of like that. And then I'll use pink for phloem tissue all around here. But the main thing I'm trying to show you here is that protostele, I'll use blue, is a solid cylinder. So there's no holes or anything in it. Like this entire thing here, I'll use red. This entire thing is solid. There's no holes or anything. Just so you can see, that's what this is trying to show you as well. See, look, solid. There's no holes or anything at all, right? Whereas when you go up towards the other types, look, there's a big hole right here, there's a big hole right here, and here it's like you're dealing with something completely different. Okay. Yeah. So then this is what you will see. This is the vascular tissue arrangement in the stem and the roots. Okay. A little bit more about the shoot system is another new term for us today, which is microphyll. So the leaves in these early vascular plants, remember, the ones that are still alive today, right? are very, very simple, and we call them a microphyll. They're very, very simple in that they have one vein, only one vein. On this picture on the left, this is the top of a lycopodium. And each of these darker things are microphylls, right? Or leaves. So the leaves are very simple. They have one vein. Just so you know, in this sense, microphyll means a small leaf with one vein, right? Later in the course, not necessarily today, but later in the course, micro will refer to something else. So keep that in mind when we do encounter that other thing. But for now, when you see microphyll, you want to think small leaf with one vein. Here you go. This is a much better way to look at the microphyll. So like, this is one microphyll. This is one microphyll. It's a close-up of basically the same plant we saw on the previous slide. This, okay. Simple, really, really simple, only one vein. If you were to do a cross-section, so if we were like do a cross-section through this one and look at it under the microscope slide, you will see the tissues that make up that leaf. And I'll try to remember that one. So you have the epidermis, right? Goes all the way around. You have the cortex, which in leaves, remember, is called the mesophyll. So that's this whole middle area. And then I forget what color I used before, but you have the vascular tissue, which in leaves is called a vein, right? And that's where your xylem and phloem are located. Also notice this, once again, right? So we saw stomatal apparatus on a sporophyte plant for the first time in the hornworts. But going forward, pretty much all plants that we look at will have these structures on them. And remember, here's an example of one here. Stomatal apparatus is our cells that help regulate gas exchange, right? Okay, we're finishing up here. We're almost done. One of the last things we gotta just talk about a little bit is the evolution of this microphyll, right? Because recall that those really early plants, right? Those early plants, like first this intermediate plant, and then our very, very first, well, based on what we've discovered so far, the first example, earliest example of a vascular plant. And then later this guy, right? The point is, these didn't have leaves, remember? No leaves. Look, this one, the intermediate plant, no leaves, right? This one, the earliest vascular plant example, no leaves. This one, the one that we later found out had the modern, right, the protostele, modern stele arrangement, no leaves, except now we have this thing called Enations. And remember that the last time that I brought this up before, look, which we believe to be the precursor to the first leaves. So going back to that, so keeping that in mind, right, now we're dealing with these early examples of vascular plants that are still alive today on our Earth. And they do have leaves, right? They do have leaves. They have microphylls. So how the heck did we end up with microphylls? Where did what events occurred for us to get microphylls on plants? And so here's what we think, based on fossil record and other things. So we think what happened is this, and this is called the Enation theory. That's what it says here on the top, right? Enation theory. So it's theorized that the way that leaves came about was that the vascular tissue in the stem over time maybe got closer and closer to the surface of the outside of the stem, but eventually kind of migrated and bent out of the stem into the enations. And over evolutionary time, once the vascular tissue entered the enations, basically serving as a vein of a leaf, you end up with microphylls. So what that might look like is something like this. So here is evolutionary time on the left. So here, this is a stem. This is a stem. Here, this is a stem. All of these things going up are stems. And then this little bud here is an enation. And so you could see this vascular tissue, right? First, in the more primitive plants, first you have just only vascular tissue in the stem. There's no leaves yet. There's no enations either. Then we have these things called enations. Vascular tissue is still not going into the enation. The enation is just this little pokey. But over evolutionary time, that vascular tissue started to migrate into the enation. Once that occurred, you basically have a microphyll, because now this structure has a vein inside of it. So that's what this is showing, right? No vein inside, enation. Once vein is inside, microphyll. Okay. Oh yes. Last thing is, so remember, I don't know if you remember, but I did say it earlier, but I'll show you again if you don't believe me. Remember when I said this earlier? Where are the sporangia located on these guys? The sporangia are located on leaves, Well, these plants only have, so far that we've learned about, microphylls. That's the only leaves they have so far, right? And so if the sporangia are on leaves, the sporangia are on microphylls. If a microphyll has a sporangia on it, we call it a sporophyll. Phyll, remember, means leaf. So microphylls is a micro or super small phyll, leaf with one vein. Sporophyll, phyll is a leaf that has what? Sporangium on it, right? Sporangium on it. We will use the term sporophyll many, many, many times going forward. So don't freak out about learning a new term today. There you go. So sporophylls are leaves that bear the sporangia, so sporophyll. Sometimes, just so you know, and we'll learn more about it when we go into the specific plants, but sometimes the sporophylls look just like a regular microphyll, and they're green and they're pokey and they're photosynthetic. And sometimes the sporophylls look a little different. If they look different, they're usually smaller than the regular microphyll. They're usually not green like the regular microphyll. And sometimes they are arranged in a new structure that's called a strobolus, which again, we will say a million times. Strobolus down here, the plural being strobili. Okay, so let's look at examples of the ones that look like regular microphylls. So the sporophylls that look like regular microphylls, and then the sporophylls that look a little different. So here first is sporophylls look just like regular microphylls. So each of these green pokies is a sporophyll. Each of these guys here, green pokey, green pokey. And I don't know if you could see it very well, but on top of each of those green pokies is this little dot, and that little dot is sporangia. Those little dots are all sporangia. But aside from the sporangia being there, the leaves themselves look like regular microphylls, right? They're small, they're green. They're not arranged in any specific way. They're just on the plant. Whereas if you look at number 2 here, the sporophylls look morphologically different than the regular microphylls. So regular microphylls are down here. Green, pokey, regular looking microphylls. The sporophylls, though, are up here. Oops, that's not the color I wanted to use. The sporophylls are here. Here's another one here, or here's another bunch of them here. Here's another bunch of them here. Here's another bunch of them there. And so here, the sporophyll, so each of these little, it's hard to see, but each of these little things on here is a sporophyll. It's hard to see. We'll see better on other slides. And notice there, each of those little things are smaller than the regular microphylls. Each of those little things, each of those little each of the little sporophylls up here are not green, right? And in the case of this species, these sporophylls are arranged in what I mentioned on the previous slide, this new structure called a strobolus. So this whole thing is a strobolus, and each of these little things on it are the sporophylls, right? Here are 3 strobili, right? Here are 3 more strobili. Here is 1 strobolus, just to start working with those new terms. This is to just show you it a little bit, because it's really hard from this to kind of get a sense of what's on what. So this is a microscope slide showing, so this one here, this one here is the same as number 1, right? We're looking at number 1 again. Notice on the top, up here, these leaves up here, they don't have, that's not a good color. These leaves up here don't have sporangia on them. They're just regular microphylls, right? Green, 1 vein, simple, right? But the leaves below, these down here, do have sporangia on them, and so we call those sporophylls. If you're going to do a close-up of a sporophyll under the microscope slide, that's what this is showing you here. This green thing down here is the sporophyll, and on top of it, this right here, maybe I'll use 2 different colors. So this is the sporophyll, oops, the green part, and then this thing that's attached to it is the sporangium. And obviously inside the sporangium is the spores. This is a better microscope slide showing the same thing. I'll use the same colors, right? So here is a sporophyll. Here is another sporophyll. On top of that sporophyll is a sporangium, right? Attached to it. Microscope slide on the right. Here is the sporophyll. And here is the sporangium attached to the sporophyll. And obviously, spores are made inside of the sporangium. Okay, so that's an overview of those extant early vascular plants, the ones that are still living on Earth. The ones we're going to cover in this course are the phylum Lycopodiophyta, which we'll talk about next class. After that, we'll talk about the phylum Equisetaephyta. And then following that lecture, we'll start talking about the ferns, which are in Polypodiophyta. And I know when I say these words, right, Lycopodiophyta, Equisetaephyta, Polypodiophyta, you're like, I'm never going to be able to say that word. You will. You will. It just takes practice. It just takes practice. And a reminder, and I'll put a reminder for this on Canvas as well, that assignment 2, which covers liverworts and hornworts, is due on Thursday before 10 a.m. before class begins. I will put a reminder on Canvas as well. If you have any questions or need clarification, feel free to write them in the discussion boards. Or if it's sensitive, you can send me an email, okay? And then next class, Thursday, we're gonna cover the lycopods. So Lycopodium and Selaginella. Okay, have a great one, guys. Thanks again. Thank you.